Close the bottom. We'll check it. What the
So good afternoon on this warm and rainy Florida morning. I want to thank John Ping and of course Chris and the whole ICBR team for allowing me the opportunity to come and present our Mission Bio solutions. I know we have some folks here live in the audience as well as some others that have joined us on Zoom. So we have microphones set up in the room so the audio should be good and we're monitoring that live. So I hope you all had a better morning than I did. I actually came out from my hotel and had the opportunity to see my passenger side rear window smashed in. So they kind of helped themselves through rummaging through my car, and I never had a problem in Gainesville. Fortunately, they left my Mission Bio literature in my notebooks, which you provide. They didn't see the value in stealing those. So there they are sitting in the back, and feel free to take some of the literature or take some of the books. So who is Mission Bio? So Mission Bio is an organization that is headquartered in South San Francisco. And in South San Francisco, hi, good morning. So in South San Francisco, we were founded out of the laboratory, Adam Abadi's laboratory at UCSF, and there was a need to kind of build upon the capabilities, next generation sequencing, and then also add some additional enhancements that would allow you to combine immunophenotyping with next generation sequencing. So Mission Bio's Tapestri is the name of the instrument, and that's what you see over here. And essentially what it is it's a very sophisticated, largely prepped instrument, and it leverages microfluidics to be able to flow individual cells through, to be able to identify mutations, as well as if you decide to do proteogenomics, as well as surface markers on the same cells. So it's the only single cell platform that allows us to do targeted and high throughput single cell DNA sequencing, and also do proteogenomic characterization, which includes on the same cells and in the same clonal populations, we have genotype and immunophenotype. We also offer the capabilities with in-house as well as field bioinformatics folks, targeted assay development to where you want to go with your research. So why is DNA and immunophenotyping useful? So immunophenotyping markers present on the cell surface of leukocytes. And they do vary with stage of maturation, so they provide a lot of information. And then you combine that with DNA and protein, as opposed to facing the same kind of constraints and limits when you have to try to combine from two different methodologies, genotyping and phenotypic information. They're on one platform, so you don't have to integrate the informatics and you don't have to make any inferences. So it gives us a better understanding of the disease, and there's myriad applications that it can be used for. Everything from characterizing the disease to identifying diagnostic and prognostic markers, but also for live cell therapeutic development and characterization, as well as post-treatment surveillance. We also can stratify patients as well. So what we were able to do is combine single cell precision to identify complex heterogeneity. So folks are used to using bulk NGS. The challenge is it's insufficient to measure clonality. There are some inherent limitations. For example, bulk sequencing is limited to 2 to 5% variant allele frequency measurement, whereas with single cell DNA sequencing using Tapestri, we can get down as low as 0.1% clonal frequency. In fact, many of our customers have even gotten higher resolution and down to lower clonal frequency as low as 0.04. The zygosity, it has to be kind of inferred or assumed to be heterozygous above sample, because all you're doing is looking at the total of the average of the tumor, which is actually a very heterogeneous sample. However, you're trying to make one kind of bulk measurement to kind of cover for a lot of different cells and a lot of different cell populations. With single cell DNA sequencing, we can definitely quantify it. Clonal architecture also has to be informatically inferred, whereas in the case of single cell DNA sequencing, it's quantifiable. Similarly, with mutation co-occurrence, that has to also be inferred, whereas it's actually quantifiable as you look at different populations of clones using single cell DNA sequencing. And with tumor samples oftentimes being contaminated with adjacent stromal tissue and other cell types, a lot of times bulk sequencing is going to be using up reads or spending reads to sequence what is essentially wild type sample. So with single cell DNA sequencing, we can actually resolve that, and folks doing studies when they might be interested in germline measurements, germline mutation and whatnot, they're able to have that information in individual clonal populations. So how does Tapestri work? I'm going to kind of walk through this a little bit slower, but your first step, and you can do a cell enrichment. Now that's not necessary. It really depends on the research questions you're trying to ask. Some folks will start with the bone marrow right away, or in some cases blood, or a solid tumor, and you don't have to do any kind of enrichment step. And then you can do the staining step. And we have a series of antibodies that are available from BioLegend, which is our partner in this, and they make a total C antibody cocktail that a lot of folks use that kind of comes off the shelf. It has a lot of the more common antibodies that folks are interested in, looking at clonal lineage and whatnot, but certainly they have at least 150 others that they can barcode to work with our system. So that's the first step. And so the Tapestri instrument, again, a sophisticated library prep instrument, it allows you to combine the protease and the sample in a matrix of oil. So you'll have the single cell will actually have be inside the oil matrix, and it'll be the combined sample and protease. You'll have the cell lysate. So that's where the DNA actually gets released. And then a second step you're going to have is where the barcoding beads and the reagent will mix together using a second oil, which is a barcoding oil. And then those barcoded cells will come out and you'll do your library prep. And it's leveraged by a 3' PCR process. So these cell barcodes enable single cell resolution. So each kind of, if you look at every individual cell with its dominant immunophenotyping, as well as the genes that it contains and the mutations that it contains, it's essentially its own sample resolve. And what these barcodes allow you to do is to bioinformatically look at these individual cells and cell populations. You know, for example, to study clonal evolution or studies subtypes or rare clones, and be able to rectify and resolve them using standard NGS. So we typically recommend 60 to 80x to get the required depth for sequencing. And we do have catalog and virtual panels. So essentially, you could go and acquire a CML or AML panel that's already ready-made. And then a lot of our customers have submitted their own panel designs and mutations that they found to be informative and actionable. And even beyond that, we can do custom panels up to 1,000 amplicons of 225 base pair each. So if you're doing a project and maybe you're interested in the content that we have available, but you want to add something, certainly that's something possible. Or you can go completely in a different alternative and design your own panel de novo. But what's really helpful here and looking at this is that these barcodes also allow cellular deconvolution. With bulk data, you would just see that aggregate of all of those different mutations, but here you're able to resolve individual cells and look at what their mutations are. So you can see co-expression of mutations as opposed to just a bulk measurement or aggregate of the mutations across the sample. So we essentially bookend the NGS workflow, and we offer a kind of soup to nuts, if you will, solution, which starts out when you're considering your experimental design, when you'll be doing your target selection. And I just mentioned that we have the pre-designed DNA panels, but also the custom DNA panels. But we have a software utility that uses in silico modeling. You can actually upload your regions of choice or whatnot, and the Tapestri designer will help you pick those amplicons that will help measure what you're looking for mutations. And typically the majority of our customers currently are doing human, but you can also do mouse or whatever the case may be, as long as the sequence is known. We do have field as well as in-house bioinformatics capabilities that would be happy to help you, particularly as you design your first panel. But a lot of people have come to me and said, wow, Tapestri Designer is really easy to use, and that's for first timers. So along with that, it also comes with, we can have the option of doing pre-designed or custom cell service from our partner, BioLegend. So the library prep, depending, typically takes 1.5 days. with 4 to 6 hours of that being your actual hands-on time, so you can do other stuff in the lab at the same time. And then the sequencing run, you use a standard Illumina sequencer, depending how many samples you want to batch or whatnot. You typically see a lot of folks use a NovaSeq if they have access to it, but the NetSeq 2000 also or 550 actually works fine as well. And the idea is to kind of, kill the flow cell and keep the sequencing costs down. But again, that's something you can work through. And then we also have a pipeline, a specific pipeline that can take hours up to 2 days, depending on how many samples you have and how big they are. We can make panels up to 1,000 amplicons, and then we have a Tapestri insights at the Tapestri pipeline, and that allows you to visualize clonal evolution, cell types, mutations, and also immunophenotyping data. You can actually produce publication quality graphs right from that software you're taking. So here's an example of a use case you might have. Again, it can work as a complement to bulk NGS in the sense where, say you have a population of patients, you would do bulk exome sequencing. So you've identified a series of mutations of interest in that population. The next step would be to design a panel, a custom panel if you go to that accommodates all of those mutations, and then do a complete single cell DNA sequencing of all of those individual patients. You'll be able to take that data that you glean from whole exome sequencing and pull it down to individual patient levels and identify different clonal populations and identify where that mutation was found along with other mutations as well. So you get a more kind of comprehensive view of that individual patient where those mutations sat. So again, it speaks to one of the points that with trying to resolve variant allele frequency in bulk exome, those were found to be overestimates that you would expect just because of some of the limitations of not being able to distinguish wild type and just a series of things where it's actually overestimated. So that kind of highlights the challenge of trying to resolve individual subclones, whereas with single cell you can actually accurately resolve all of those individual subclones. And then we also wanted to make sure that the same mutations would be measured from bulk exome sequencing and with doing single cell, and we found that there was a very high correlation. In this graph here depicts how tight the coefficients of variation now with having the same kind of mutations able to be measured both on the panel and in the bulk, but then also the added value that you get at being able to identify subclones and be able to resolve the mutations more accurately. So we do have leadership in a few key applications. Again, we're emerging. I'll speak to a few of the things we're going to be focusing a little bit more on going forward as well. But historically, hematology has been and always will be kind of our core focus. We actually have 3 catalog panels, and then as I mentioned, the published and the virtual panels, but we also have solid tumor capabilities. But we have one catalog panel, which is a tumor hotspot panel. And essentially what's coded on there is the ability to detect some of the more common oncology mutations that are found across different solid tumor types. And we also have 2 virtual panels, one of them for glioblastoma and one for breast cancer. And again, with those, you can look at the content and say, this is great, but I want to add a few other things. And certainly we could do, our content plus and make you a custom panel that includes everything that you're looking for. And for solid tumor, we typically use a universal nuclei isolation protocol. And S2 Genomics has been our partner as well, and they have a nice system and protocol or whatnot to be able to get nuclei to be able to be flowed across the system. And then we have custom panels for characterizing specific targets and for genome editing and for mouse models. So we really have a very flexible system, and as long as the sequence is known, we can definitely work together to build something that picks up the mutations that you're interested in. So we sort of alluded to the fact how complex cancer is. It's not one homogeneous aggregate, whether it be a blood sample or whether it be a solid tumor, but rather it's made up of a whole bunch of different clones, a whole bunch of different populations, and these populations mutate and they change over time. They change as a function of time, and they also change as a function of the tumor cells trying to develop mutations or find ways around the therapeutic or the chemotherapy that they're being treated with, whether it be a targeted therapeutic or more common chemotherapy medicine. So what ends up happening is you end up getting a very different distribution of types of clones and identification of clones than when you started. As you have at the very beginning, you have pre-treatment that works all the way up to going across different targeted therapeutics. And what's important to note here is that at relapse you have a very different clonal map on the fish plot of saying that there's very different clones existing at that point. And one could infer that maybe the treatment itself is selecting for or causing the accelerated growth of certain clones that weren't necessarily there in the beginning, but you're also seeing, because of the benefits of targeted therapeutics, the certain clones that maybe they were targeting disappear and are becoming certainly less common. So we can use single cell proteogenomics pretty much across the entire patient journey. And I'll certainly go into a little bit more point that we're at some of these points where our investigator customers have actually used and answered key questions using single cell proteogenomics. So one option is to detect high risk precursors. So a lot of times that, when you're doing bulk sequences, you're probably going to miss this because it's sort of lost in the noise because it's a low abundance clone. But some of these precursors can actually be the clones later on that end up causing a problem. After you've gone through the treatment paradigm, they can be responsible for drug resistance, they could be responsible for remission. So it just kind of kinetically, as the cancer evolves and changes both on its own and as a result of being treated with specific therapeutics, you're able to look at these to potentially some of these high-risk precursors and maybe treat them a priori early on, recognizing that they may become a problem later on. So also it offers the opportunity for stratification. If you want to stratify different patients, or you want to look at a series of different clones and say, what would be my optimum therapeutic regimen, maybe to target specific mutations that are already manifest or are beginning to emerge. And also if you're designing a trial or whatnot, you might want to look at how to stratify patients and maybe select for those who are more likely to benefit from given therapy. And you can also monitor response, and you can identify those rare subclones as you're surveying and your therapeutic is taking hold and the patient is pretty stable. You can look and see, well, any of those dangerous clones appear and then potentially be a problem later. We could get ahead of it. So another thing that's caught sort of our interest quite a bit lately, and we've demonstrated, our customers have demonstrated the ability to use, is characterizing measurable residual disease, particularly in AML, but also we have investigators working across ALL and other hematological malignancies to be able to more accurately characterize MRD versus the current methodologies they may be using. And also we can ID distinct relapse and find out what exactly was causing relapse and maybe get ahead of it before it becomes a problem. So one, and again, I apologize that it's kind of up here with the little Zoom blocks up there, but essentially what Dr. Nato and Dr. Campo did is they were able to look into Richter transformation. And Richter transformation is a particularly poor outcome typically, and it only appears in about 5 to 10% of patients with CLL. And what typically happens is we really don't know the mechanisms or the changes that cause someone to go from the CLL to Richter's transformation. But what is known is that it typically occurs within or after the first few months of treatment. So one could maybe wonder if the treatment itself is having some kind of a factor in the etiology and onset of Richter's transformation. So what this group wanted to do is figure out if single cell sequencing can resolve that clonal heterogeneity, but also detect any rare pre-existing clones or mutations that might portend a later progression to Richter's transformation. So what they were able to find, and this publication is available on our website, we have links to all of them, that single cell DNA-seq was able to identify some of these rare subclones at the time of CLL diagnosis, and these were actually dormant from anywhere from 6 to 19 years. So imagine that. So those clones are sitting there lying in wait for just the right opportunity, whether the patient's weakened for some reason, or maybe the treatment is selecting for it. But then all of a sudden they rapidly expand and transform, and the patient develops the more nefarious Richter's transformation subtype. And the findings suggest that potentially we could do early intervention to maybe identify some of those dominant clones and eradicate them early on and design a treatment regimen that would target those before they become a problem. And then essentially you might be able to avert or at least greatly delay the onset of Richter's transformation. So if you compare against the information you would get from bulk NGS, and this speaks to a different study, and this one was McMahon et al. In cancer research, you can follow variant allele frequencies for bulk NGS, but it doesn't give you the whole story. So what's essentially happening during this is you're seeing this patients being treated with gilteritinib, and you can see a downregulation or reduction in the amount of clones that actually have the FLT3. But you also see an increase in NRAS. But again, this is all bulk, it's all in the aggregate, so you don't know exactly which mutations are found on which cells with each other, or you don't even know exactly how many clones there are to carry each one. All you can see is that the bulk, assuming that the clone is present in a high enough proportion, that you're able to detect it. And what's interesting here is it's NRAS1 at day 57. It's virtually undetectable, but you see it begin to rise. So similarly, what you would expect with a FLT3 mutation, you would expect gilteritinib to effectively minimize it. So you do see that it falls off and begins to reduce. But what's really interesting here is if you follow this with single cell sequencing, you actually see these populations that you're not able to resolve here when you look at the bulk, and you see there's a quadruple mutant, though it does have FLT3, which one would think would make it susceptible to the targeted therapeutic. It actually has that NRAS mutation that we've seen pick up that they're able to start seeing at day 57. They're able to see, and we're able to resolve that one becomes a problem, as well as the double mutants, which actually does not share or have any kind of a FLT3 mutation. So we have two things. We have a treatment-resistant clone, and then we also have a target-naive clone. And both of those are problems. As you can see, compared to when the disease was diagnosed, they become the dominant clones, and they're responsible for the relapse, and they're going to present a new challenge as the physician decides a treatment regimen going forward. But what's also very important to look at is from the individual clones, here we're able to resolve 3 cells out of the whole population, and 89 cells, which would basically give us even less than that 0.1% of rare clones that we're able to detect. But we got down to 0.04% using Tapestri, and then down to 0.8%. Imagine if we had this information that these clones would be getting to emerge here at this early stage, rather than waiting until it got to the point where, maybe bulk would identify to this patient, hey, there's a new mutation that came up. And imagine if you can combine and figure out exactly the individual genotypes of specific clones, and recognize that you were trying to design a strategy to treat a very specific population, rather than kind of dealing with the incomplete information that you have involved. So similarly, these Dr. Masali and Dr. Rosen also wanted to study in solid tumors, how does resistance happen? So they used combinatorial KRAS G12C and EGFR inhibitors for treating CRC, but one of the challenges is emergent resistance. What is the cause of it? So they seek to kind of look forward and go into looking at individual clones and clonal populations to try to figure out what was going on. So what they were able to identify is that KRAS G12C amplification is responsible for resistance by engaging the senescence program. And then also that single cell sequencing revealed that the NRAS G12D mutation occurred in the same cells bearing the KRAS G12C alteration. So you kind of had a troublesome clone, and that was causing resistance they were observing. Okay, so this is just a graph from Dr. Chris Horrigan's lab, and it shows you the kind of information, or the depth of the detail of information. This one happens to be an AML sample, but you realize you can do even more detailed and deep classification. But with just CNV alone, you basically have kind of 2 that you would be resolving it into. But when you go with combined protein with chromosomal abnormalities, as well as with the copy gain and loss, you're able to resolve very specific populations and be able to look at the frequency of these genes in the population and give you a little bit more detailed information as to what's going on in this individual patient, versus if you just had bulk and tried to interpret flow data in parallel with it. So you're able to actually combine both in the same platform. So we alluded earlier to detecting minimum or measurable residual disease, and we've had a lot of interest in that using our platform. And Ross Levine's lab at Memorial Sloan Kettering, they've been using Tapestri, and they've been able to identify it with a high sensitivity and high specificity, as opposed to what's typically been used, which is flow cytometry or our QRT-PCR, and then some people use bulk sequencing. But again, you're looking at a bunch of methodologies and you're trying to cobble together what happens when they don't agree. For example, you get a situation where a patient comes up as MRD negative with flow, but yet NGS says they're positive, or vice versa. How do you manage that indeterminate or imprecise phenotype? Well, it would help to have a single technology or a single platform that would give you the answer to that. So what Ross Levine's lab wanted to know is can this single cell MRD assay detect these MRD clones at a sensitivity, specificity that's better than current methods? And from what is seen here in the preprint and what they've observed is that they got down to 10 to the negative 4. Current flow MRD is 10 to the negative 3 as the current standard. Some labs can even get to a higher resolution, but typically the baseline or what is expected is 10 to the negative 3. And in addition to that, they can also resolve the clonal architecture. So not only are you seeing is there MRD and being able to make a call within reason, but you're also to see the individual clones and clonal populations in that individual patient. And you also get immunophenotype data too. So not only are you able to find the mutations in these clones, but you can also see cell lineage or any kind of phenotypic information that might be informative as well. And then another great thing too is we're able to separate out clonal hematopoiesis. With an older population, sometimes you start having a collection of clonal hematopoiesis genes and cells, and some types of technologies may infer those to be leukemic when they're not. And there's a lot of studying going on to try to see what its role is and what makes them maybe potentially switch to become leukemic. But you certainly want to resolve that and determine which the cells are, as opposed to just like to call everyone leukemia or overlooking something that, well, I think it's clonal hematopoiesis, but I'm not really sure. So being able to have that high detailed clonal information, I think would support having a little bit more actionable insights. And again, those weren't able to be found in bulk NGS, but they were actually present at relapse. So here's an example of some different mutations that were missed by bulk. A lot of times it's because, again, they don't have the same resolution to get down to low abundance because you're sequencing everything. So something that might be relatively low allele frequency is not going to be detected in the noise because you're sequencing a whole lot of wild type and a whole lot of other things. So it's only those most prominent genes that are going to be identified. But these were actually also present at relapse. So I would argue that they were pretty informative, maybe making future treatment decisions. But also using the immunophenotype data, we can find those specific patient populations and differentiate CHIP versus leukemic clones. So again, having the combined genotyping and immunophenotyping information was helping you be further able to resolve than what you would do if you just did bulk NGS or flow cytometry. Okay, so another take home that we're able to look at post-genetic stem cell transplants, so post-transplant patients, they were able to resolve donor and host pairs, which is really fascinating. And also to take a look at those same samples and see distinct cell surface protein expression and resolve. So if you had CHIP clones, you could identify whether they came from the host or whether they actually came from the patient themselves. So just having that combined genotyping data and very, very high sensitivity along with the immunophenotype provided a lot of additional potentially actionable information. And it's key to note that it's definitely more sensitive than even bulk single tandem repeat quantitation. So more MRD monitoring with AML. Oftentimes, the thing we talked about with it being a little bit challenging to figure out between pre-leukemic clonal hematopoiesis and malignant clones. Well, can we use single cell genotyping to detect those patterns? And then the second question they had is clonal diversity, or the amount of different clonal populations that a given patient has predicted for relapse. And as we saw with some of Ross's work also, the group at Stanford also unambiguously differentiated clonal hematopoiesis from leukemic clones, but also they found that decreasing clonal diversity or fewer types of different populations of clones were also associated with a longer relapse-free survival. And intuitively, that might make sense because the more different populations of clones that you have floating around, the more combinations of different mutations, and the more you're almost playing whack-a-mole with those different types of clones, trying to find either a targeted therapeutic or a chemotherapeutic strategy to knock them down. So if you have more of them to deal with, it would intuitively look like you might have a little bit more of a challenge in eradicating them. So it sets up a good model during remission to take a look at measurable residual disease. We used 3 different time points, and what they actually found is that at these different time points, they could see whether a patient was going to go in the direction of actually going towards a relapse based on the clones they have, or actually into remission. So again, you could envision a paradigm where a diagnosis and remission and relapse, you use single cell sequencing, if not necessarily like at bulk NGS at diagnosis and relapse, but also in the middle during remission, just to kind of chronicle or monitor, if you will, potentially the emergence of clones that might portend a relapse as opposed to the more benign remission phenotype. So these are 3 different patients here, and these are essentially the fish plots that show a few different things of what can happen. So first one actually shows that if you have high sensitivity, you can detect that disease relapse clone right at remission. It's the same quadruple mutant clone at diagnosis, remission, and relapse, but kind of interesting when the patient begins to relapse, how that clone becomes once again the dominant clone. So something has happened to allow that clone to take over. And then similarly, you can have a different clone at remission and relapse than you do at diagnosis, whether it's the treatment or just some kind of internal mutation for the cancer trying to survive or whatnot, but you end up with a different clone. Now here in the third case, you actually have relapse clones that are able to be distinguished from chip clones. So you can use single cell data to be able to determine, okay, these aren't leukemia clones, but these are chip clones. And again, that's being able to take individual clonal populations and doing full proteogenomics with immunophenotyping and single cell DNA will allow you to have that additional information to help you distinguish that. It's interesting to follow that, you know, these are 3 different patients, and they all follow somewhat of a different trajectory as they go from, you know, initial diagnosis all the way down to remission. But having the highest resolution opportunity to be able to pull up these low abundance clones is valuable because it kind of gives you an early access insight into what's going on and what's going to be the problem later on. So it could suggest that people could actually model what's going to happen moving forward and design their therapeutic strategies accordingly. So taking a look at this take-home point, you have a similar degree of clonal richness at diagnosis, but low clonal diversity in remission would be longer relapse-free survival. And then assessment of MRD status and AML can help inform escalation or de-escalation therapeutic strategies, but it may also be important going forward for clinical trials. We kind of take a look at the directions things can go in. Well, if you see CHIP or MRD negative, you can go to consolidation treating this patient. But if the patient is MRD positive, you may go with escalation as opposed to going for a stem cell transplant. And then MRD positive with these clones, you could end up with a targeted therapy against a mutation. And this is going to minimize the likelihood of over-treatment, but also minimize the likelihood of maybe missing something and not treating the patient who might be at high risk aggressively to be able to head something off. You know, as opposed to not having such a high resolution measurement, you might have to kind of do a little more waiting and seeing. So we would think that this would potentially support better insights to support better patient care. So another area of interest that's new and emerging for us, but using Tapestri technology, is measuring genome editing. So why would you want to use single cell for cell and genome therapies? Well, it allows you to measure multiple genotypic and phenotypic parameters in individual cells, so it gives you a more comprehensive characterization of the ex vivo cell therapies that you're developing. Because we know, one of the things that needs to be measured is transduction and transfection efficiency. We can look at vector copy number. You can see whether that vector is integrated or whether it remains episomal. You can also check using a CRISPR. You can see if you have on-target or off-target modifications and edits. You can also identify chromosomal aberrations. And if you're doing multiplex edits, it allows you to see how efficiently, sometimes you'll have 1 or 2 that'll take just 1, and it allows you to figure out exactly what your distribution of cells is with a given mutation and how many can be there at the same time. So you may get all 6, you may get 5, the combination of 5 that you get may differ. So you're essentially being able to monitor your entire population at the clonal level and see the individual edits and how efficiently they're transformed. Similarly, you can also measure zygosity, which is also not able to be resolved with bulk. So here's one example of measuring gene transfer in cell therapies. Using Tapestri, you can measure transduction, transfection efficiency, as well as measure individual vector copy numbers. And this graph shows just an example of a project that was done collaboratively with a customer, where you're able to resolve the copy number. So unlike bulk, where you get population averages, you're able to actually resolve individually the amount of gene transfer in here. So oftentimes you want to see what the vector copy number is. Folks will set a limit, like I don't want anything more than 5 or 4 or whatever the case may be, for safety reasons. So this allows you to unambiguously resolve individual populations and see how well your bulk therapeutic is working based upon individual cell populations. So similarly, you can look at gene editing. So for therapies modified by CRISPR, Tapestri can measure on and off editing in each cell, and that on and off editing is measured by the percent of indels. So multiplex editing, zygosity, and translocations can be assessed in contrast to traditional bulk, where you would lose a lot of this because this would all be lost in the averages. But here, as you can see, for example, with EHBB, 26% of it is off target. Similarly with CLTA, 74% is on target, but you actually see 0.08%. And again, it's that high resolution of different clonal populations that allows you to get that level of resolution to pull out those individual populations. So you're actually seeing data from individual cells as opposed to being subject to bulk averages. Okay, and translocations. In this case, they use 3 genes, HPV, CLTA, and RAB11A. They co-edited using CRISPR and Cas9, and they actually were able to identify translocation events with high sensitivity down as low as 0.2 to 1.8% of cells. So that's pretty high resolution. And again, you wouldn't see these events. You would just get a bulk average if you just use bulk sequencing. And similarly, we were able to resolve into these different cell types or clone types, and you're able to identify the amount of edits, but not only the amount of edits, but also the zygosity. So looking across here, these are all the different combinations you could have, and it actually gave you the amount of cells that carried the individual endowments. You had several that had 1, 2, 3, 4, 5, 6 edits, and in this case, 6 edits, and they all became a homozygous edit. But you can resolve and break down and characterize that production and see how many of each type and how much mutation and the zygosity in those individual cell populations. So taking this one step further, the Dana-Farber Cancer Institute, they wanted to see if they did co-mutations, if they did multiplex CRISPR edits, can they analyze those at single cell resolutions? They chose 6 particular genes. And again, their question wasn't to determine what these edits or these changes in these individual genes, what downstream effect they have, but rather they wanted to see if single cell immunophenotyping was able to detect those different edits and resolve them. So what they actually found out is that CRISPR-mediated on and off target editing, which we saw a couple of slides earlier. So not all your edits are going to be on target, but also zygosity, and you're able to measure it at the single cell level. So, and then finally they took a multiplex edit itself, they were able to transplant them into mice, and in the mice's body, you'll see that they were selected based upon the combination of edits that they had. So this is essentially their workflow, and they had targeted these 6 specific genes. And as you can see from the graph below, this gives the number of cells for each gene edit combo. So this is the intersection size, so how many picked up each mutation. And then this is the number of cells with edits at the target. And you can see there's just a myriad of different mutations that could be found in a given cell. And this is the amount that would have just 1 mutation. And then, but yet you would have 1 edit And this one here would actually show 2 edits, 3 edits, 4 edits. You have a very diverse population. But again, in bulk sequence, it'd be very difficult to recapitulate this and identify it. But using individual populations, not only to be able to resolve the number of cells for each gene edit combo, but you're also able to see what cells actually have the target, see how efficiently a given gene is edited. So in this case too, there was only one combo of mutations that remained at high frequency. They put them in pre-transplant, and actually putting them into the animal's body, there was only a selection of cells with MGA and CHD2 co-mutants. And just going back, we have that particular action, so CHD and MGH carried over. So that individual type of cell population actually had the 2 edits combined, where it was actually able to be resolved in the post-transplant mouse. So all of those other populations were not competitive for survival. So that's what I've got, and if there's any questions, I'm happy to answer them.
Is this just the target approach, or you can do whole genome?
It's targeted. It's targeted, so we can go up to 1,000 different amplicons, and they're about 225 base pair. And again, you would use our Tapestri designer software, and you'd be able to identify and build your own panel or use something off the shelf. We had already designed it, or one of our investigators or one of our customers had designed something and provided the content.
So how many cells you can target? So analyze it.
We typically recommend, if you're going to do full immunophenotyping, you start with 1 million cells. But if you're doing full genotyping, you start with 100,000. And our capture rate right now is about 5 to 10% of everything that's flowed across it. We're working on what's called our generation 3 chemistry, and that will pull that level up. So if you start with 100,000 cells, you'll probably capture about 5 to 10 on a given experiment right now. And we expect that to improve with some of the enhancements that we've made to the assay.
So what's the 53? So double the rates.
Okay, so we call it version 3. So essentially, it's going to increase, potentially double it to about 20%. But again, there'll be experiment to experiment variation depending on your cells, that sort of a thing. So, but you could say that you get right now, currently with the current chemistry, somewhere between 5 and 12%. But we do expect that to improve a little bit and get a higher percentage of capture. But certainly, even capturing 10,000 cells gives you the opportunity to pull up a lot of low abundance cells and rare cell populations. Yes, sir.
Hi, I have a couple questions. The first, what type of gel beads do you use?
Okay, so the beads that come into the reagent kit, so everything comes, we give a list with the
What type
reagents.
of beads?
They're manufactured for us by a third party.
It's a hybrid gel bead or different?
I think that's what they're made out of.
Okay. The second question is, compared to the 10x genomics,
Yes.
a platform, what are the advantage and disadvantage of your?
Okay, so 10x is a great platform. They do gene expression. We do DNA and protein.
Those DNA and protein on the same single cell?
Yes, yes. So we can do the combined DNA and protein on the same cell. So full proteogenomics, whereas what they focus on is gene expression profiling primarily. So it's not necessarily a replacement, but it's an adjunct, and it's a different way to look at cells, for different information. So a lot of people who have one have the other. We find it just depends on the question they're trying to answer, which works best for them. But they both kind of, work in tandem.
Besides the DNA and protein, you platform applicable to the RNA as well?
We currently don't do RNA. We've had customers ask about it, but right now it's not something that we commercially support. Again, as you probably saw inside of those droplets, everything is in there. Everything is in there, but we don't have a supported assay to derive RNA. But it's in there. Yes, sir.
So you just mentioned for the protein and DNA that's connect together, or you just Well, from the single cell, you get two kinds of information or just.
Yes, so as long as you have viable cells, you can decide, like some folks do an assay and they're only interested in the DNA and I'll do a DNA only assay. But then you can do, if you want to look at protein, there's just that additional step up front where you stain the cells.
From the same DNA.
Yes, but it'll be the same cells and you'll be able to resolve individual cells. Look at not only their mutational status, but also the immunophenotyping or the proteins that are present on those cells. So it kind of gives you both.
Does the system select for those cells that are positive for the, yes, for the proteins that you're interested in? Or are all the cells stained and then routed?
The latter. The latter. So some folks may foment and design an antibody selection strategy. They might not all want all 45 that are around our cocktail, but they may want to use very specific individual ones. But what it will show you is where it's which ones have stained positive, which ones have stained negative, to be able to break out those populations based on the markers that you're interested in. The informatics, and again, they'll be barcoded, so you'll be able to see, you know, cell 3,140 is stained positive for these ones and not, you know, these others. So it kind of gives a combined opportunity, but it flows everything.
And it's 45 targets is the maximum for protein?
That's what our off-the-shelf panel is. We have about 160 that are available. I don't know if anyone has pushed it to try to use all 60 yet or anything, but I don't think that that's a limitation. I think it's just the 45 or so that people are most interested in. And we wanted to have something off the shelf to be able to bring to customers so they didn't have to go through the customization or whatnot. So we have that panel and we find it covers a lot of more common applications. But working, you know, with BioLegend and whatnot, they have, you know, at least again up to 150 that you can mix and match and make kind of your own cocktail. Thank you. So, anything else? Any other questions? Want to buy a Tapestri? All right, thanks, everybody. Help yourself get the looted books and everything.
Dr. Lick's lab. I'll see anybody who's Dr. Lick's lab.
Okay. Okay, yes, okay, okay. Please, if anybody who has questions, I can send it to you or not. It'll be good. Thank you, I appreciate it. It depends on the question you're trying to answer. That's the tough part of it. Like if you wanted just a very specific cell population, I would do an enrichment with maybe 900 beads. Because other than that, we'll be looking at like different kinds of cells that might not necessarily be of interest for you. So if you want to be able to get a specific population, I would start with enrichment, because in that way, then it makes sure all the measurements you got are on cells of your type that you were interested in. But we do have people that want to see everything and they flow. Okay. So it just really depends on what your question is.
In that way, you can see the positive and negative
Yes.
populations.
Yeah.
And how it works is the first time there's a flow.
Yeah, I can actually do that. Yeah.
I didn't get the part how you label the cell.
Yeah, you can remember, that's a-
Part and then you label with the particle and then you
Yeah.
label them with the applicant.
Yeah. Yeah. You want
Hey, Eric.
to go all the way back and just help with me. Look at that when I look back.
Hey, Eric, can you hear me?
Yes.
We had one question actually in the chat. So once you're done answering this, I can pass that on to you.
Okay, awesome. Thank you.
Oh. Do you know the estimate that price for this sample? How
It goes in that screen. Yeah, no, it goes about 2,000 to 3,000 a sample. If it's 3,000, you're doing the full immunophenotype, but about 2,000 if you're doing just the beginning, you get a ton of information. You get all these different cell populations and mutational status. I always say compared to what though?
If you bring patients for like the one that we use, we used to tell them how that smaller market location when we want to, because if they have the, minimal residency.
Okay.
So that will be beyond our target. We're.
Yes. I know what we're working on right now, being able to multiplex for the MRD assay. Well, that's what we could probably potentially do. That's what we're working on, the MRD assay. That's about that. Still. Is that AML? Is it AML?
Yes.
This one?
Well, keep on page, but we don't know who have the main thing we're going to do.
You can multiplex and create ones with the new MRD. So you can but it also will get a lot of people with 2-3 patients at once, which will bring that sample cost down a little bit by their 2 nerves.
Yeah, we want to propose for a random application, but with that price range, I don't think.
And again, it's all the more, if you do 150 samples too, it's gonna come down like that. That's the base, that's the list, that's, you know, 4 samples. So you can benefit from 2 things, multiplex them on the MRD assay, one, the second part of the fall.
So the 2002, contain the sequencing part of the library.
Yeah, your sequencing cost would be whatever you work out with the board here at UF. They have a difference, so it'd be just enough to get to the library.
Oh, that's the library part.
And that's that'd work into it again. That's reagents, that's the panel. So there's a lot there. But
If we skip the cell surface,
just for genotyping,
yes.
that's a little bit less, yeah, because it's about 3,000 with both. It's 2,000 with just the DNA. But our MRD assay that we're developing actually has 3 years due antibody hashing to be able to combine 3 samples together and be able to individually resolve the patient. So,
Okay, for one library,
yeah, I wanted to do that. And again, that meets the, you know, because we know from clinical utility, we're gonna need to be able to meet a certain price point. So that's why we're gonna be able to do exactly what you said. So we'll be able to do that. But essentially how it works is you blend a sample and protease together, and then it comes in the oil, and then there's the sample with the protease, slice the cells and all the protein and everything is inside here. And we barcode it, so each barcode is each individual cell. Use barcoding oil, and then that moves to your 3 PCR triple primer, PCR library prep.
So this PCR steps do
Yes.
will contain all the barcode.
Yes.
Earlier,
Yes.
no matter which team you're interested in, they.
Yeah, it's gonna resolve it. Yes, because it's targeted, so every cell will be immunophenotyped and will be sequenced. So you'll get, but it's targeted, so it's not doing like whole genome sequencing in each cell. It's just the targeted genes that are in the panel, those are the protein. So it's an awesome upfront. So why don't you follow up and we can discuss a little bit more about the MRD. How do I get a hold of you?
It's okay. We actually that's the that we want.
Okay.
For a grant, for viewers, maybe because the cost, you know, for the library distribution is.
Sure. But it's better to stick with targeted, and sequencing costs for what you want to see. Versus sequencing everything, whole ends up, you know, a whole bunch of a whole bunch of sequencing stuff and necessarily action.
Right.
Thank you.
Did you sign in?
So Eric,
Okay, good. And
you know, the chat up there at the top. So Melanie has a couple questions. She could come in Tapestri.
Make sure you know about it. I think this is really great. And anyways, I work in ICBR.
Oh.
Yeah.
Hello?
Hi.
I have a couple of questions.
Sure.
Okay. I'm already using the Tapestri platform, and I
Great.
wanted to know why you changed your barcoding oil, and was there a reason behind it? Because I didn't see any issue with the way it was used before, but now you're having, like, it has a requirement to store at 4 degrees, and I'm just wondering what changed.
Yes, we always want to have continuous improvement. We found that in some customers' hands, as the oil got older, it looked a little bit cloudy, and we just wanted to take a little bit of the variation out of there and get something that would be more standardized and be able to last the full duration of 6 months plus. So we added a specific additive in the oil to keep it fresher for longer. And we've also found in our studies that it kept much better when stored at 4, and you would get a longer shelf life out of it.
Okay,
You have a Tapestri?
yes, I do. Yes. The second question I have has to do with the antibody washes. Over the course of the last 2 years that we've used this system, we have had significant loss in cells with the antibody washes. And I noticed that the more recent protocol has increased the number of cells,
Yes.
but I'm still finding that there is a significant cell loss. So I'm wondering, do you have any sort of recommendations for how that can be rectified based on the current protocol, and if you have any new improvements over the last 6 months?
Okay, so I haven't heard any complaints for that particular issue. One thing I would ask you is how old are your reagents?
Well, the reagents that we're currently using are approaching, like, they're probably like 20 months old.
Okay, all right.
So
So we typically don't like to go any longer than 12 months for the reagents. So that could be one of the reasons. What I would suggest is we get on a call with FAS Sean and kind of talk through it and maybe share some data and just kind of get a look at what you're observing. And then maybe identify a path forward. But a lot of times the aging of the reagents beyond the shelf life can be an impediment to having the most optimal or efficient binding.
Okay, well, this is something that we have seen over the course of the last 2 years, even before the reagents were past the 12-month shelf life. So, but of course, that was with a previous protocol. So with the new protocol, we have not been able to try this with more recent reagents, but if for some reason, because we do have some reagents that we purchased recently that we can test this with to see if we still have the same amount of cell loss. But if I still continue to see that, is there any other recommendation you might have?
Yeah, I would say we get on a call with Sean and kind of talk through it. Would be if this batch doesn't work, and then I would kind of look, because we haven't seen that as being a challenge in the field. So I'd certainly want to get ahead of it and see if there's anything we can do, because I'm sure we can, figure something out to optimize the results you're getting. But with those newer reagents, I'm curious to see exactly how things work out.
Okay, because I've talked with Sean about this for years, and
Yes.
I mean, he even came to our laboratory to even do an actual extraction with us, and he saw right in front of his very eyes the same problem.
Okay, and you still, we don't, okay, so why don't we get together and just try to approach it maybe a little bit of a different way? Let's get a call with them, because I want to make sure you guys are back up and running to, meet your standards.
Okay. I mean, let me go ahead and try some of, I think when Sean came, the reagents were maybe at the 12-month mark then.
Okay.
So, so I would be kind of inclined to maybe try it with some of the reagents that we currently have, just to see if that truly is the issue before actually moving forward, considering we've had this issue for years.
For sure, yeah, let's just find out what the problem is and what we can do to get you back on track. But Sean is definitely the guy, so let's try to schedule something and work on it. I'd be curious to see what your results with the different batch of reagents are.
Okay.
Anything
So, there is one other question I have. I'm sorry. So is there any other, so in addition to the barcoding oil, I know you have changed the protein protocol a little bit. Is there any additional reagents in the newer kits, like thinking thinking anything after April of 2021. Is there anything in the newer kits that may actually increase the efficiency of Tapestri as opposed to anything in the older kits?
There aren't any changes, but stay tuned because in the next couple of months, we're gonna be announcing a few things that may increase your capture rate. But that's
Okay.
not necessarily ready for prime time yet, but in the next several months, we expect something that could potentially increase the amount of cells you get versus the 5 to 12% that you're seeing now.
Okay, perfect. Thank you.
And again, Sean will be the first one to be a conduit to be able to access that. But I recommend we at least try those different reagents that you have and see if we can improve the situation you have. And if not, we definitely should get on a call with Sean and see how we can work through this.
I agree. Thank you. I appreciate that.
Thank you, Melanie.